talking about DNA replication, and more specifically, the prokaryotic replication fork. So, what is the prokaryotic replication fork, and what even is DNA replication? Well, basically, all DNA replication is just making more DNA. And it's going to be occurring typically in our nucleus if we're a eukaryotic cell, but in this case, we're prokaryotic, which doesn't have a nucleus. But still, prokaryotic cells are going to be doing different types of DNA replication, with just some differences from eukaryotic. Now, where is this going to be happening, this whole replication thing? Well, it's going to be happening over here in our origin. So the origin is basically going to be denoting the area of active replication, which is going to be called the replication fork. So we know that whenever any active replication is going to be going on, that's what our fork is going to be. So in order to understand the complexity of DNA replication in eukaryotes, scientists then needed to look into prokaryotic replication first, such as E. coli, in order to then establish and understand this replication fork, and then get our enzymes coming into play. Now, our key enzymes that we need to look into and pay attention to are going to be our DNA gyrase, our SSB proteins, helicase, primase, DNA polymerase 1 and 3, and our ligase. So, first of all, we have helicase coming in here and unzipping our DNA strands. And it's doing this unzipping through breaking the hydrogen bonds between all of the bases. So helicase is coming in here, opening up our replication fork, and DNA gyrase is going to be back here, unwinding our strands and basically locking down these two strands so that there's no supercoiling happening. And supercoiling, though it sounds super, it's actually not in this case, because we don't need anything to coil, we need everything to stay unwound in order for our enzymes to properly encode. So gyrase does the job of locking things down and keeping things in place and not winding back up. So that's DNA gyrase and our helicase. Now we have our SSBs, which, if you can see, this just means a single-stranded bonding protein. And now these bonding proteins are coming in here, and they are the ones who are going to be locking the strand in place so they don't come back together, because we don't need them to come back together, we need to copy them. So our proteins are going to be hanging out, making sure that nothing's going to be sticking back together, and we need to stay open. And so then we have our primase. Now, primase is really important because it makes our RNA primers. And these primers, I drew them in green because these are basically like the start area for our DNA polymerase. It's going to be our little star area or our green light or green flag saying, hey, start coding here, because DNA polymerase needs that guidance to be like, okay, where am I going to begin to copy? And that's where our RNA primer is. And our DNA polymerase is going to be doing that transcription of changing our RNA to DNA as we're going along these fragments. Now, in this case, these fragments are down here in our lagging strand due to the fact that these are needing to be read in the 5' to 3' direction, but in this case, it's being read 3 to 5 over and over, which is making the discontinuous pattern. Unlike the leading strand, which is easily being read from our 5 to 3, and having our continuous pattern, because down here we have our fragments breaking up because it's having to keep up with our leading strand and having to pick up and go and pick up and go, causing for this lovely gap in the middle. Now this gap is going to be filled in by our enzyme of ligase, which is basically the gluer of this bottom lagging strand, because we need to still have this continuous pattern even though our DNA polymerase wasn't able to do that properly. So we have our ligase coming in there, gluing everything together. Our primase is the leader saying, hey, start here. The DNA polymerase is picking up 1 and 3. And the main difference between DNA polymerase 1 and 3 is basically that this DNA polymerase is the one that's going to be the proofreader, and our number 3 is going to be the one that's basically picking up and going and making sure that these primers are turning into RNA. And that is all you need to know for our replication fork.